So I'd like to thank the organizers for fitting me in this wonderful lineup of talks. As you will see now, this is something completely different. Okay, so my talk is titled Coordinated Regulation by lncRNAs Results in Tight lncRNA-target coupling. So since its inception about 10 years ago, my lab has been interested in non-coding RNAs, and in particular also long non-coding RNA regulation. And our workhorse for predicting long RNA targets is called Longhorn. It was published in multiple papers, including the ones that are listed here. And Longhorn combines sequence or motif-based evidence and model-based evidence to predict long non-coding RNA targets. Models are listed here: switch, decoy, cofactor, guide. I'll just give you a bit of a flavor of a couple of them. So switch long non-coding RNAs are long non-coding RNAs that bind promoters to inhibit or activate transcription factors, or 3' UTRs, other parts of the RNAs to inhibit and activate microRNAs, RNA binding proteins. To find these, we look for sequence evidence, and then we mine expression data to see evidence for the model, for the presence of these types of interactions. Cofactor long non-coding RNAs only regulate transcription. They bind proximal promoters and either interfere or recruit transcription factors. So we've been looking at long non-coding RNA regulation for quite a while, and I've been interested in studying how well we do and improving the accuracy. And more recently, we started categorizing long non-coding RNAs as either cytoplasmic, nuclear, or those long non-coding RNAs that are present in good abundance in both the cytoplasm and the nucleus. And so naturally, we wanted to see how well we do in terms of predicting targets for long non-coding RNAs from each class. Looking at cytoplasmic long non-coding RNAs, what I'd like to show is that our prediction on the right does much better than sequence alone or for non-specific predictions. What we were concerned about was that when we looked at nuclear long non-coding RNAs, we saw that none of our models make any difference. So the benefit of Longhorn has been almost entirely post-transcriptional. And so we started to debug this, trying to understand why we are not improving on sequence alone, sequence motif alone, when predicting long non-coding RNA regulation of transcription. And what we became convinced of is the reason is that the sequence-based observation that is based on triplex rules has too many false positives and too many false negatives. And so in a paper that is now posted on bioRxiv and is under review, we try to improve that. And we designed a method we call now Bighorn, that instead of using triplex rules for RNA-DNA interactions, it uses kind of a fuzzy system of connected gapped k-mers to model long non-coding RNA binding sites. In doing that, combining this model instead of triplex with our model of regulation, we were able to significantly improve on predicting long non-coding RNA regulation of mRNA transcription. So what I'm showing here is silencing experiments of 6 long non-coding RNAs, and this is just in one cell line. The red is Bighorn, everybody else is in the bottom. Bighorn does much better. We were actually so impressed with our ability to improve transcriptional regulation that we started comparing targets of each long non-coding RNA that we studied. And the surprising thing was that we found for many long non-coding RNAs that they appear to regulate the same gene transcriptionally and post-transcriptionally. In a sense, you can think of it as regulating transcription and then chaperoning the product or the message all the way to its translation. So what this slide shows, on the left, transcriptional interactions only by long non-coding RNAs, their count and our ability to verify them. In the middle, post-transcriptional interactions. And on the right, interactions that we believe are coordinated, where the long non-coding RNA regulates its target both transcriptionally and post-transcriptionally. Coordinated targets are many, and our ability to verify them is much better. When we looked at these interactions, we focused on long non-coding RNAs that are expressed highly across many cells and that are dysregulated in cancer, and looked at their coordinated targets. And one sort of jumped at us in particular. ZFAS1 has all of those properties, but one of its targets was predicted to be dicer1. And we studied non-encoding RNAs, so we thought regulating dicer1 is kind of cool. So we went and silenced ZFAS1 in multiple, in cell lines for multiple tumor types, and checked to see if dicer1 really moved. And it did. It's up there. There are others, but it's up there. So we started studying this particular interaction. We silenced ZFAS1 in multiple cell lines for multiple tumor types so that dicer1 RNA is down. Protein was down even further. We showed that ZFAS1 affects promoter activity and 3' UTR activity of dicer1. And we showed that targeting ZFAS1 and dicer1 lead to very similar effects on the miRNA. The way we think of this is this. Dicer1 is a target of microRNAs, and it also regulates microRNAs. They live in some sort of a steady state equilibrium. Changing ZFAS1 expression changes the steady state equilibrium. By changing it, we can either upregulate dicer1 in the miRNA or downregulate it. Moreover, phenotypes that are known for dicer1 can be phenomimicked by targeting ZFAS1, including cell proliferation, response to radiation, and targeting ZFAS1 also affects tumor growth. My acknowledgements, and I think I'm good. Okay.
Hi, Pavel. Oops. Gonna take this. This doesn't really work, right? It is on. Okay. Really cool stuff. I really think we need to just better understand what these lncRNAs do. And so you mentioned that you incorporated like Hamer rules to somehow like predict triplex interactions better, if I understand. I mean, you didn't really explain. Could you say more? Because last year we published a paper.
We're limited in time. You mean this stuff?
Okay. How exactly did you do this? I mean,
so the draft is posted on the bioarchive. I could spend another 10 minutes talking about it. So we can talk about it after if you like.
Okay.
Yeah. Yes.
Yes. This might be a bit outside the scope of what we presented with before, but I wanted to know from the transcripts that have that you can tell they have an effect on regulation, how many can you not directly link with the target? Like you think the long non-coding is involved, maybe it's in the vicinity like this, but there's no direct sequence interaction.
Yeah. So I don't remember frankly, but one of these models tries to sort of, and so the idea is that the model generates properties, but then we can go and mine data to sort of see if the properties fit. And we've spent a lot of time sort of trying to verify, or estimate the accuracy that we are able to capture these models. But I mean, this was done over like 32 tumor contexts and developmental data. All the numbers are going to be different, and I don't remember them. Sorry. Yeah.
ZFAS1 is a host gene for SNO-RNAs. When you knock it down, do you know if you're affecting the abundance of SNO-RNAs? And
I'm sure you do.
maybe more generally, do you see a difference for your long non-coding RNAs who are hosts for other non-coding RNAs versus the ones that don't?
Yeah, so we have a draft in sort of we're working on to sort of do something similar to what you're describing, but I don't have the data with me. The reality, I mean, I think of RNAs in a cell as like just a bunch of floating like things, and that every change you make is going to affect them in all kinds of ways. But I agree that there are a lot of hosts, but you have so many. I could draw a map of just ZFS1 and dicer. It would be like 12 different arrows pointing to different things and changes, right? The regulatory system is very complex. But yeah, you're right. I'm sure that that's happening too. All right, thank you.